Welcome students. Today we will be discussing a very important short note, that is transamination reactions. So how you define transamination? Transamination is a process in which there is transfer of amino group from one amino acid, one alpha amino acid to one keto acid, alpha keto acid, and resulting in formation of new alpha amino acid and alpha keto acid. Thus, there is interconversion of a pair of amino acid and a pair of keto acid. So, the transfer of an amino group from one amino acid to another keto acid is known as transamination. The reaction involves interconversion of pair of amino acids and a pair of keto acids. The overall process is catalyzed by a group of enzymes known as transaminases or aminotransferases. The derivative of vitamin B6, that is pyridoxine phosphate, acts as coenzyme. So you can see this reaction. So amino acid is converted into one amino acid is converted into one keto acid, and keto acid is converted into amino acid. So in pyridoxine phosphate is coenzyme. So transamination is a reversible process. Most amino acids except lysine, methionine, proline and hydroxyproline undergo transamination. Not only amino acids but guanidino group of ornithine also undergoes transamination. So where is the site of transamination reactions? The main sites are liver, kidney, brain and heart, but it may occur in most of the tissues. Enzymes. Which are the enzymes? The reaction is catalyzed by transaminases, also called aminotransferases, and they require pyridoxal phosphatase coenzyme as it forms essential part of active site of enzyme. So specific transaminases exist for each pair of amino acid and keto acid. More important transaminases are SGOT and SGPT. SGOT means serum glutamate oxaloacetate transaminase, and SGPT it means serum glutamate pyruvate transaminase. So these two enzymes are also of diagnostic importance and are also called AST and ALT respectively. AST means SGOT. AST stands for aspartate aminotransferase and ALT means alanine aminotransferase, SGPT. Mechanism. So what is mechanism of these reactions? So transamination exhibits ping pong mechanism of enzyme action. And occurs in two phases. In first phase, alpha amino group of amino acid 1 forms a sheaf base with aldehyde group of enzyme linked pyridoxal phosphate. By sheaf of double bond, aldehyde is converted to ketimine, which is hydrolyzed to release alpha keto acid 1 as a product, leaving pyridoxamine phosphate held non-covalently to the enzyme. This is first phase. In second phase, alpha keto acid 2 binds with pyridoxamine phosphate and forms ketimine, which is changed to aldehyde, which is cleaved to release amino acid 2 and regenerating enzyme linked pyridoxal phosphate. So alpha amino acid 1 is converted into alpha keto acid 1. Pyridoxal phosphate is converted into pyridoxamine phosphate. Then pyridoxamine phosphate combines with alpha keto acid 2 to form alpha amino acid 2 and pyridoxal phosphate. So what is importance of transamination reactions? So it redistributes amino nitrogen amongst amino acids. It plays a role in catabolism of amino acids as well as synthesis of non-essential amino acids. Third is it produces glucogenic products such as pyruvate, oxaloacetate and alpha ketoglutarate. So these are the three important functions. So again in this figure it is shown alanine in presence of alanine transaminase. So glutamate plus adenine in presence of alanine transaminase, it forms pyruvate and alpha ketoglutarate. this is reaction showing aspartate. So aspartate plus glutamate, it forms oxaloacetate plus alpha ketoglutarate. So this is mechanism of transamination, reactions catalyzed by ALT and AST, and formation of sheaf base is shown in between. So sheaf base, I will again revise. So transamination exhibits ping pong mechanism and occurs in two phases. In first phase, alpha amino group of amino acid 1. So alpha amino group of amino acid 1 forms a sheaf base with aldehyde group of pyridoxal phosphate. By sheaf of double bond, aldehyde is converted into ketimine, which is hydrolyzed to release alpha ketoacid 1, leaving pyridoxamine phosphate. So aspartate combines with pyridoxal phosphate to form pyridoxamine phosphate and oxaloacetate. So oxaloacetate is ketoacid which is formed. So in second phase, alpha ketoacid 2 binds with pyridoxamine phosphate. Alpha ketoacid 2, that is alpha ketoglutaric acid, it will bind to pyridoxamine phosphate and forms ketimine, and which is changed to aldehyde, which is cleaved to release amino acid 2, that is glutamic acid, and regenerating enzyme linked pyridoxal phosphate. So alpha amino acid 1 is converted into alpha ketoacid 1, pyridoxal phosphate pyridoxamine phosphate is converted to pyridoxal phosphate, then alpha amino acid 2 is converted into alpha ketoacid 2. So that is the